Assalamu alaikum wa rahmatullahi wa barakatuh. In this lesson we will be discussing osmosis. At the end of this video, students will be able to define diffusion and osmosis, define hypotonic and hypertonic media, and determine what happens to an animal cell when placed in a hypotonic, hypertonic, and isotonic solution. To accomplish the objectives of this lesson, we will need to follow up in worksheet number 3. What happens if you add an ink drop to a cup of water? The ink drop will spread into the whole cup of water. Do you know what we call this phenomenon? It's called diffusion. To better understand diffusion, we prepare a container having two solutions separated by a semi-permeable membrane. One solution has high concentration, the other has low concentration. Due to the difference in concentrations, the solute particles will move from the medium of high concentration to the medium of low concentration throughout the membrane until equilibrium is attained. That is, we get the same concentration on both sides of the membrane. Knowing that the animal cell has a semi-permeable membrane, how does diffusion occur at the level of a cell? In a cell, water always moves to reach an equal concentration on both sides of the membrane. Diffusion of water across the membrane is called osmosis. If the cell is placed in a solution where the concentration inside the cell in the intracellular medium is equal to the concentration outside the cell, the extracellular medium, then the two solutions are said to be isotonic, and water molecules will move into and out of the cell at the same rate, and the cells will keep their normal shape. And if the cell is placed in a solution where the concentration outside the cell is less than the concentration inside the cell, the solution outside the cell is said to be hypotonic, and the extracellular medium will be richer in water than inside. Then water molecules will move into the cell, and the cell swells and may burst. However, if the cell is placed in a solution where the concentration outside the cell is greater than the concentration inside the cell, the solution outside the cell is said to be hypertonic, and the extracellular medium will be poorer in water molecules. Then water molecules will move to the outside of the cell, and the cell will shrink. Now let's have a brief evaluation and match each state of the cell to the corresponding type of solution it's placed in and the observed effect. The first image shows that the cell shrinks. Then it's placed in a hypertonic solution. The second image shows that the cell is normal. Then it's placed in an isotonic solution. And the last image shows that the cell swells. Then it's placed in a hypotonic solution. Another application. Knowing that the concentration of sodium chloride in the cytoplasm of a red blood cell is 0.9%, explain what will happen if the red blood cells are placed in a solution where the percentage of sodium chloride is 0.13%. To explain this case, we have first to compare the concentrations of the solute between the intracellular and extracellular media, infer the type of solution the cell is placed in, describe the direction of water movement, and finally describe the state of the cell. Then we will get this answer. The concentration of sodium chloride in the extracellular medium is higher than that the intracellular one. Then the cell is placed in a hypertonic solution where water leaves the cell and the cell shrinks. Thanks for listening.